For all of human history, we've accepted aging as a simple fact of life. But what if we started to see it differently? Not as an inevitability, but one of biology's most complex puzzles. Now I know that sounds pretty wild, but I'm Mick, and in today's video we're exploring the fascinating and sometimes unbelievable science that is starting to ask, what if the steady decline of our cells isn't as fixed as we thought? Imagine a future where our cells could be reverted back into a more youthful state, where the very hallmarks of aging could effectively be reversed. Sounds like the premise of a pretty neat science fiction movie, right? Well, thanks to the collaboration between OpenAI and Retro Biosciences, that future is rapidly becoming a scientific reality. So let's get into it. But first, let's get into who we're talking about. We have, of course, OpenAI, a leader in artificial intelligence and well known for their GPT models. OpenAI has joined forces with Retro Biosciences, which is a biotech startup focused on expanding the human lifespan. Retro Biosciences actually got their initial investment from Sam Altman, who is, of course, the founder of OpenAI. Their shared belief that AI can meaningfully accelerate life science innovation. And to put this belief to the ultimate test, they embarked on a groundbreaking project, using a specialized AI model to enhance something truly remarkable called the Yamanaka factors. So, what are the Yamanaka factors and why are they so important? The Yamanaka factors, specifically a set of genes including OCT4, SOX2, KLF4, and MYC, often abbreviated as OSKM, were discovered by the Nobel Prize-winning scientist Shinya Yamanaka. These factors that the genes translate to have the ability to reprogram adult cells into induced pluripotent stem cells, which are known as iPSCs, turning back the cellular clock. This cellular reprogram has actually led to the development of potential therapeutics for conditions like blindness, diabetes, and even addressing the organ shortage crisis. It's completely changed the field of regenerative biotechnology. But there are a few challenges that we have to consider. The traditional reprogramming process is quite inefficient. Typically less than 0.1% of cells convert, and it can take up to 3 weeks or more. This efficiency drops even further in cells from aged or diseased donors, making optimization a critical research focus. Plus, directly optimizing these protein sequences is incredibly hard. For instance, SOX2 alone has over 300 amino acids, and the number of possible variants is so fast that traditional methods can barely scratch the surface. This is where OpenAI comes in. They designed and trained an experimental custom model called GPT-4b Micro. Think of it as a miniature version of the powerful GPT-4o, but specifically trained for protein engineering. It was given a broad base of knowledge in biotechnology and trained on a massive data set including protein sequences, biological text, and even tokenized 3D structural data. What's amazing is that a large portion of this data was enriched with contextual information, which allows GPT-4b Micro to be prompted to generate sequences with specific desired properties. This is a huge leap. It enables it to handle complex proteins like the Yamanaka factors, whose activity depends on flexible, unstructured regions. The model truly represents a shift in AI-guided protein design. So let's go ahead and get into the results. By leveraging GPT-4b Micro, Retro Biosciences was able to design novel and significantly enhanced variants of the Yamanaka factors, which they've called Retro SOX and Retro KLF. In laboratory experiments, these redesigned proteins achieved greater than a 50-fold higher expression of stem cell reprogramming markers than the wild-type controls. These wild-type controls being, of course, the standard Yamanaka factors. But yes, you did hear that right. 50 times more effective. The model's suggestions were incredibly potent. Over 30% of the Retro SOX sequences outperformed the wild-type SOX2, even though they differed than more than 100 amino acids on average. To put this in perspective, traditional screenings typically see hit rates below 10%. That's compared to 30%. For KLF4, the model delivered even better results, with nearly 50% of the generated retro KLF variants providing turnovers. But we're not done yet. When the top retro SOX and retro KLF variants were combined, they produced dramatic gains in both early and late pluripotency markers, with late markers appearing days sooner than the standard Yamanaka cocktail. These findings aren't just isolated observations. They've been validated by replication across multiple donors, cell types, and delivery methods, confirming full pluripotency and genomic stability in the derived iPSC lines. They even used mRNA delivery and mesenchymal stromal cells from middle-aged human donors, achieving over 30% expression of the key markers in just 7 days. The resulting iPSCs could differentiate into all 3 primary germ layers, showing healthy karyotypes and genomic stability suitable for cell therapies. But it gets even better. The team investigated the rejuvenation potential of these re-engineered variants, specifically their ability to restore youthful characteristics to aged cells. They focused on DNA damage, which is a key hallmark of aging. And what they found is just crazy. Cells treated with the retro SOX or KLF cocktail showed visibly less YH2AX intensity, which is a marker of double-stranded breaks because DNA is double-stranded, compared to cells reprogrammed with the standard OSKM or control. This suggests that these AI-designed variants are more effective at reducing DNA damage than the original Yamanaka factors. By helping resolve these core hallmarks of cellular aging, these engineered variants offer a potential path towards improved cell rejuvenation and use in future therapies. Just in general, this work is a powerful illustration of how quickly a domain-specific AI model can deliver breakthrough results on a focused scientific problem. As Boris Power, who leads research partnerships at OpenAI, puts it, when researchers bring deep domain insight to our language model tooling, problems that once took years can shift to days. Retro Biosciences is now looking to implement these newly engineered Yamanaka factors in longevity experiments, including assessing their ability to prolong the lifespan of mice more effectively than the original factors, while of course carefully monitoring for any potential risks like tumor growth. And it is definitely important to note that GPT-4b Micro was developed for research purposes and is not broadly available. And as I did mention before, we have to consider Sam Altman, the CEO of OpenAI, is a huge investor in Retro Biosciences. But imagine a future where AI-guided protein design helps us unlock deeper insights into aging and develop therapies that not only treat diseases, but also extend human lifespan. The possibilities are boundless as they are thought-provoking, and are definitely going to lead to a lot of ethical conversations along the way. And on that note, I'm going to have to ask you, what do you think about the potential of AI in life sciences and age reversal technology? Share your thoughts in the comments below. And if you found this video interesting, make sure you like the video and subscribe for interesting science that I want to share. I'll see you in the next video.